today I will be talking a little bit about the breeding efforts that we are currently doing in order to develop new cultivars particularly for legumes for the Southeastern United States. So this presentation will build a little bit on some of the points that Dr. Quisenberry mentioned last week. For those of you that don't know, Dr. Quisenberry is still active and working with us. He works with me and with Dr. Kevin Kenworthy, who is a turf breeder. So we are very fortunate to have Dr. Q still around and providing mentoring not only to me but also to my students and postdocs. So hopefully I will cover some of the points that you came to look for, and I will be happy to answer questions at the end of the presentation. So we have a very diverse livestock industry in the state. We have a very large number of beef cattle operations, but also very important and very large dairy operation in the state. They may have different requirements in terms of the needs for forages. And I've been very fortunate to, as Jose mentioned, to hit the ground running with projects. But those projects have not been possible without the support from the Florida Cattlemen Association and also from the Florida dairy farmers. So I really appreciate their support to the breeding program. Some of our forages also serve as a wildlife habitat, and I think Marcelo will cover some of that later in the series with the pollination efforts and some other ecosystem services. Also, we have a very diverse environment in Florida. We tend to think that Florida is hot and humid, but in fact, we have very distinct environments, not only in terms of temperature or temperature changes. In winter, particularly in the Panhandle, we can have a significant number of frost or even freezes, or sometimes even some conditions for snow. So that creates some particular environment in that region, while South Florida is considered primarily tropical. So the species that we work with hopefully will be adapted to these two major regions, but some of them will be able to grow as well in the whole state. We also have various soil types. With that, we are targeting specific legumes, particularly for the flatwood soils, and other legumes that are well adapted to more well-drained soils, such as alfalfa and crimson clover. Dr. Dubé asked me to present this, or to make the presentation, particularly focusing on the last part of the breeding program, basically in the testing and release of cultivars. I will still want to touch a few basic aspects for the early part of the breeding program, and I will show you a little bit why I would like to do that. So this slide was modified from Dr. Boughton's slide that he presented in my class a few years ago in terms of forage breeding pipeline. So we first need to set up our breeding goals, and I will show you what are my breeding goals in terms of what are the species and what are the target traits that we are working with in our breeding program. We then have to screen for germplasm and have genetic diversity in our breeding program in order to be able to continue and expand the breeding efforts into the future. Then it comes the part of breeding and selection, and depending on the species that you're working with, you can have self or cross-pollinated species, and then different selection or breeding methods. And lastly, we're going to focus primarily on testing and releases, and I will share with you some of the cultivar releases in the last few years and what is coming up next in the market. Also, I would like to, I often tend to think that I'm just a forage breeder, but I also have to think that I'm working in a public breeding program and in academia, and we have to fulfill the mission of the university in terms of education, research, and extension. So besides creating cultivars, I'm also here mentoring students and mentoring postdocs into forage breeding in order to be able to continue and pass some of these information to the future generations. In terms of defining the breeding goals in my breeding program, we are working with cool and warm season legume species. In terms of cool season, I will present a lot of information regarding alfalfa. as well as some of the work that we are doing on white, red, and crimson clover. I will briefly touch some of the work that we're doing with warm season legumes such as cowpea, achinomene, hairy indigo, and Alice clover. I'm particularly interested in yield. All of you will agree that yield is the #1 trait that we should focus on, but not only yield in terms of total production, but also in terms of distribution and when do we need that yield in order to feed our animals or our livestock. We are also looking at nutritive value and a couple of biotic stresses. I will show you some results for nematode tolerance that we have done in a number of species. And we recently started working more on abiotic stresses, particularly on drought and also looking at fertilizer applications. I put together this slide because of some of the questions that I saw last week when Dr. Q finished his presentation. Some of you were asking, why are we not breeding Sanghem? Why are we not breeding some other species? And I mentioned that having genetic variation and having genetic diversity is very important for our breeding program. So this table was created by David Boswick. He was my first graduate student in my program. So I asked him to basically summarize some of the information for some of the key legumes that could be used as cover crop or forages in terms of warm season and cool season. I'm not going to bore you with all of the information in this table. I just want to highlight 2 species. So Sanghem here. Look at the number of plant introductions or accessions, or look at the genetic diversity if you think for Sanghem in the United States. In the USDA, there are only 13 plant introductions in the system. So there is no much genetic variability for me to justify running a breeding program when we don't have genetic diversity for a given species. However, look at cowpea. Cowpea is one, besides soybean, is one of the warm season legumes that will have one of the major or largest germplasm collections in not only in the United States with more than 6,500 accessions, but also in Africa and IATA, the germplasm collection has more than 15,000 different cowpeas. That means that we have enough or very broad genetic diversity from where we could select and improve our forages. With that, I will just give you one example of why having genetic diversity is important. We're working with these large collections, and I will show you one example for one of the traits that we were looking for diversity. And I think I started showing you some of the results with Melodogyne incognita. This y-axis represents the reproduction index. It's basically the ability of these nematodes to grow in the roots and reproduce themselves inside the roots of these nematode, of these cowpea plants. As we can see, there is a large number here in the left, all these genotypes, including iron clay, which is here, are showing excellent resistance to Melodogyne incognita. All these genotypes here in the right side, in the red, in the dark red, are basically susceptible. Now, there is a new emerging nematode pest in the Southeastern United States. It's called guava root dot nematode. Maybe some of you have heard about this before, but it's a new, I would call it a new emerging pest in our regions. It's affecting several different crops, including sweet potato and tomato and peppers. It has been reported in several places in Florida, but also in other places in the Southeastern United States. When we use this nematode and we screen the same genotypes, now the graph looks totally different. We see that many of these genotypes that we tested before that were highly resistant to Melodogyne incognita are now highly susceptible to this new nematode. And there is only a very few, basically 4 accessions from this germplasm collection that are showing excellent resistance to this new nematode pest. So with this brief example, I wanted to highlight why it's important to have a very broad genetic diversity and work with germplasm collections in order to introduce these gene and these genotypes that are showing resistance or are basically that are resilient to these new emerging pests and in the future as well, new abiotic stresses. So basically the question is, do you want to grow a cowpea as a cover crop or as a forage that will basically have nematodes in the roots, or would you like to grow a cowpea that will show excellent nematode resistance, particularly if you're using this as a cover crop in your vegetable operation or basically or in your field corn operations as well. So let's go a little bit more into breeding. So once we identify good genetic material as parental lines, we cross them, then we put those families or those plants in the field, and then we phenotype them. My program is heavily focused on phenotyping for biomass production and nutritive value. I was very fortunate to come into this job, and the university basically provided the funds to purchase a new plot combine. So we are now collecting biomass production basically in all our species. And we are also running a lot of nutritive value analysis in the breeding lines using NIRS. More recently, with my graduate students, we are now collecting data using drones, basically looking at the regrowth and the biomass production throughout the year by utilizing drones and artificial intelligence as well. At the genotype level, we are using molecular markers in order to speed up the process for developing cultivars, particularly for perennial species, and also to make selections more efficiently. But let's jump into the testing because I think this is the topic that most of you might be interested in. So I will start with alfalfa. I'm not starting a new program in alfalfa. There was a breeding program in alfalfa 40, 50, 30 years ago. There have been a couple of releases. The first one was Florida 66, then Florida 77, and the last cultivar that was released from the University of Florida is Florida 99. Unfortunately, this cultivar is not commercially available anymore. So that's why we basically wanted to resume or restart the breeding program for alfalfa. Some of the examples or some of the historical yield for some of these old cultivars, we see a big jump in yield from the 70s to the 80s, basically through the use of Florida 77 and later Florida 99. And these cultivars have been developed or selected for higher biomass production. If you don't believe there was alfalfa in Florida, this picture is showing a very good or pretty good stand of alfalfa in Hillsborough County, Florida, showing excellent adaptation, particularly when we have material that was bred under our conditions. Again, the breeding program was dropped in the late 90s, and that's why we restarted some of the efforts to develop new cultivars for our region. Some of the work that we did with Dr. José Dubé in Mariana was look at some of these cultivars and management practices. In this table, we're seeing two different stubble height, 2 and 4 inches, and then 4 different varieties. As we can see, Florida 99 in general yields better than the other cultivars. And with 2 inches, we are getting a little bit more yield. However, at the end of the experiment, basically after 2 years of data collection, for those plots that were mowed at 2 inches, we see a significant reduction in persistence. So we see a lower number of plants per square meter under the treatment at 2 inches compared to the 4 inches. And if you cannot picture that in that graph, you can see this picture that José took. Basically, the different cultivars, the 2-inch treatment, and looking at particularly Florida 99 with really good stand persistence, even after a number of harvests at 2 inches. So this is showing me that we have really good germplasm as the base in order to restart a new breeding program. Some of the results from my work at the Plant Science Research and Education Unit in Citra, Florida, we tested 30 different cultivars during 3 years. So we started collecting data in March 2015 and we ended in the summer of 2017. When we look at the graph in the top, we see a seasonal production. Basically, alfalfa yield will be highest at this time of the year, starting from late February until probably late May is when you will see the highest peak of production. And these are averages by harvest in the y-axis. When we look at the cultivar performance, we see this breeding line, UF alfalfa persistent 2015. This is our most advanced breeding line that was reselected out of Florida 99 for vigor. And this is the line that we are thinking that we're gonna be releasing this year in the fall. This line produced higher yields in each harvest, even compared to Florida 99 and then Bulldog 805, which is one of our standards for our region. Some of you may like to see total yield production per year. This is coming from the same experiment. Again, in the first year of production, we tend to see that all cultivars perform similarly because there's of course no issues with persistence. But after the second year or starting in the second year and even at the third, that's when we see that cultivars that are bred and developed under Florida conditions, they perform much better and yield more compared to other cultivars. And then most of these are, again, commercial standards that you can buy in any seed store in the Southeastern United States. Hopefully this breeding line, again, will be proposed for release. Hopefully we'll go through the process at the university level, and we will be looking for a seed company to basically license this material in the upcoming year. For you to have an idea how the plots look like after harvesting several years. Oh, sorry. We also have some data from Oklahoma, and this was in collaboration with Mike Trammell. We see that this line, UF 2015, out yields Bulldog 805 and Alpha Grace in 2017 and also in 2018 in South Oklahoma as well. So we believe that it will have some good adaptation into the whole Southeastern United States. It is now currently being grown in Mississippi, and it will enter the variety trials in Kentucky as well in the upcoming years. So how the plots look like in the first year, everything looks good. Everything has a good stand. But after 1 or 2 years of harvest, not everything looks good. So here in the left, again, we have our breeding material compared to any other cultivar out there. We see that we have a much better persistent in these breeding populations. This is another picture showing excellent persistence in our program. And I'm talking about the alfalfa, not talking about the people in the program or the people in the picture. So in this case, we have Dr. Yolanda Lopez, my biological scientist, very impressed by the persistence in our line, and Dr. Q here standing next to it in a plot where it was supposed to be alfalfa, and now we have weeds. So we are very excited and looking forward to this release and to the future. Finally, I would like to thank the Florida dairy farmers and the Florida Milk Checkoff, because now we are testing this potential variety in North Florida Holsteins. We're looking forward to the results for some of these on-farm trials for this breeding line from our breeding program. So the future looks promising. We started making selections after introducing material and cultivars from other parts of the world into Florida. We make crosses, and now we are testing these lines under a lot of different conditions at the Plant Science Research and Education Center in Citra. We are collecting data at the ground level, but we are also collecting data using remote sensing to, again, estimate biomass production more efficiently. The good message here is that our new families, our new breeding materials are out yielding the controls. So we hope that we will be able to keep improving yield and persistence in alfalfa for Florida and also for other regions in the Southeastern United States. Finally, let's move to some of the other legumes. We have been working with white clover, particularly in collaboration with Jose, doing some variety testing or variety trials, particularly looking at some of our new breeding lines. We have a multi-leaf population that performs really well in Afro and also in Mariana. This year it was included again in the trials, and hopefully after the trials conducted this year in Afro and in Mariana, we will be proposing the release of this new breeding line. In terms of quality, there's no doubt that clovers have excellent quality, high protein, and digestibility. And we will be looking forward to the release of a new white clover line in the upcoming years. In terms of red clover, Dr. Quisenberry presented last week about the work that he did developing this population that is tolerant, I would say, to 2,4-D. This is the FL 2,4-D resistant population. He presented some of this data already, but here in the bottom, you can see the dates. And here in red is this breeding line or this cultivar. It flowers much earlier and produces earlier compared to Barduro, Southern and other cultivars as well. This breeding line was basically licensed by Grassland, Oregon, and they decided that the name of the variety will be Q Red Clover. So we hope that in the fall 2021, there will be some Q Red Clover in the market available for planting. And if not, for sure it will be in the fall 2022. So be on the lookout for this new variety from our program called Q Red Clover. More into red clover, we have developed a new breeding population that is tetraploid. We basically have used colchicine and double the chromosome numbers. And this is a variety trial that we did in Citra, Florida. And this is in kilograms per hectare in production per year. This line, the Florida 4X, is the Florida tetraploid population. It out-yielded most of the other cultivars, even though it was not statistically significant compared to Red Ace, Barduro, and the Florida 2,4-D red clover. We see here that in monoculture, it does very well, but it does also perform really, really well in combination or in association with other warm season grasses, such as Bermuda grass in this picture here in the top and also in the bottom. So we're looking forward to this breeding line as well. It's coming up in the pipeline. The only issue might be seed production at the tetraploid level because the flowers are a little bit bigger. And we see that there might be some issues with pollination as well. So we might have to do a little bit more work in terms of pollination and seed production in this breeding line if we want to see it in the market. Finally, Dr. Q also mentioned some work that we are doing with crimson clover last week. This was, I would say this was the first selection that I made when I started this job. In the spring 2017, we grew a population and I selected for early flowering. And this was one of the first variety trials that we ran in Citra in 2019. This was March 12. You can see clearly that this breeding line is flowering way earlier than Dixie, Sunrise, and Flame. It produced decent yields and very similar yields to Dixie, but I think the major benefit for this breeding line is the early flowering. Look at the picture that we took almost 2 weeks later after harvesting. So we basically harvested this plot at the time of peak flowering. There is no much regrowth here. So as from a forage point of view, you might be disappointed, but I'm looking at this line particularly as a cover crop because this March 21 is when most of the producers may want to start planting corn in Florida or in South Georgia. So I think this line will have a lot of impact as a cover crop in the Southeastern United States, particularly because of the early flowering and the reduced regrowth after harvesting at peak flowering. We are running again in 2021 variety trials in Mariana and at the Agronomy Forage Research Unit in Hague. We don't have a lot of data to share because we are just harvesting these plots. I can share that in terms of flowering, our breeding line, the crimson clover, was the earliest flowering. And we also are testing a ball clover that Dr. Blount was developing her program. So we are looking forward to developing a ball clover as well for Florida and for the Southeastern United States. With that, let's jump to warm season legumes. As I showed you earlier, I've been working a little bit on cowpeas. There are 3 lines from the USDA that I'm particularly interested. They are called, or they are named 1136, 1137, and 1138. They have shown, particularly 36 and 38, a much greater biomass production compared to Adam Clay and compared to ACE. ACE is a new forage type cowpea developed by Texas A&M. I think they released it in the fall 2019. And we purchased some seed last year to include it in our breeding trial, in our variety trials. We also used 3 different planting dates. We planted at the beginning of August, the end of August, and also at the beginning of September. Our idea was to try to push production towards the end of the fall, so around Thanksgiving and early December. That's when we basically harvested these plots. The first planting date, we see a significant increase in yield in 36 and 38 compared to iron clay, and also in the second planting dates. In general, none of these cowpeas like planting dates in September, so this is probably something that we would not be recommending in the future in order to push production again later in the fall. Cowpea will be a great addition to our portfolio of cultivars for forages, given the really good nutritive value. And also we are looking forward to use cowpea as cover crop as well. Finally, in terms of hairy indigo, we've been working on a project funded by SARE, looking at reducing hard-seededness in order to develop hairy indigo as a cover crop, particularly for crop rotations with strawberry. We are now under the second cycle of selection, and we are making progress in terms of reducing hard seed in hairy indigo, but I don't see any issues with using some of these breeding materials for forages as well. And finally, this is the last slide. We started some breeding efforts last year with Achinomene, also other populations for hairy indigo and Alice clover. We increased seed last year, and we're gonna be testing biomass production in the summer this year. So this is very exciting news in terms of introducing new species into our program. And in terms of releasing and disseminating cultivars, I'm actively involved in this process, but I'm also very grateful to be working with these amazing teams, not only at the university level in Florida, but also in collaboration with Georgia, Alabama, Mississippi, and other extension programs in the Southeastern United States. So I will be looking forward to even more collaborations in the future. With that, I would like to highlight that even though this is a legume conference, I released my first cultivar last year. We released New World Bermuda grass. So if you're looking forward to planting new materials for Bermuda grass, please contact me and I will be happy to share some of the planting material with you. With that, I would like to thank all the lab members, all my collaborators, and the funding agencies that make forage breeding at Florida possible. Thank you.
Okay, we have a couple questions that have come in. How long does it take from release to available commercial seed? What's kind of the normal timeline from release to commercial seed?
That is a good question. That really depends on how much breeder seed do you have. There are a couple of examples that I'm not going to go into detail, but there are examples where we didn't have enough breeder seed. So when we make the cultivar release, the company then has to spend 2, 3, or 4 years increasing seed to be commercially available. I would say that with this alfalfa line that I would like to release, I took the effort and the investment in increasing seed. And we have about 500 pounds of breeder seed sitting in our cooler and ready for the company that will license the variety to take it commercially. So we are hoping that that time between releasing and being commercially available will be reduced. And that's why we decided to increase the seed ourselves. But that really depends on the company and how much breeder seed we give to the company.
All right, the next question is, when you're developing legumes, are you evaluating their competitiveness with grasses?
That's
their ability to coexist.
Right. We have not done a lot of work yet. Or breeding evaluations yet. We have made selections of plants that have been growing with Bermuda grass or Bahia grass and then use those plants in crosses, thinking that they may have some advantage and some better adaptation to growing in combination with grasses. But part of my efforts into using drones and into using some of these novel technologies is to start doing some breeding in association with grasses. I think we are in the legume conference and we are appreciating the benefits of legumes, but I don't forget that grasses and perennial grasses are the backbone of our livestock industry in the Southeastern United States. So I think that the way to go will be to start breeding legumes that will be well adapted to growing with either Bahia grass, Bermuda grass, or Limpo grass, or any of the other perennial grasses.
All right, the next question relates to ball clover. Ball clover is very persistent across the Panhandle and Southwest Alabama and Georgia. What is your view and thoughts about developing ball clover lines?
Well, this ball clover that I show you that we are testing, this was developed by Dr. Blount or selected by Dr. Blount. I think the good thing about ball clover will be, it will fit the niche and those environments where in general we have a little bit more water and perhaps some problems with standing water even, where some perhaps other clovers may not persist very well. So since I was not the original breeder, I would say if Anne would like to chime in and give a little bit of a background for that breeding line that she developed. But I would say this is an effort with her and with Jose as well.
All right. Next question is, how about the early crimson clover? What is your expected release time?
That is a good question. In general, I will make a comment about the process for release. So we need to show at least 2 years or 2 locations, and that is the minimum amount of data that we need to show to the UF cultivar release committee. So again, I made my first selection in 2017. We started increasing seed, and now we ran 2 or 3 years of variety trials. This year we included this line in Mariana and also is in the University of Tennessee in their cover crop breeding trial. So hopefully by the end of this season, I will have enough data to propose the release in the fall of this year. But hopefully it will be either fall of 2021 or spring of 2022.
All right, next question has to do with your alfalfa breeding program. Stephen wants to know if you're looking at the below ground traits, the root morphology and the symbiosis to help with problems in alfalfa pastures.
I think these are two great questions. First is the short answer is no, we haven't done any work below ground. I think there is a lot of work that needs to be done, particularly in the sense of fixation and looking at and see if our legumes and particularly if alfalfa is fixing or if it's fixing enough nitrogen. So the short answer is no, but we are looking forward to collaborating and doing some more work with folks that are in the area of microbiology, and then also in the sense of nutrient cycling in order to understand better and measure whether our alfalfas are fixing nitrogen. I think Jose mentioned last week or he made a comment regarding the use of nitrogen in alfalfa. I would say that we have done that, we have tested that, and that really helps, particularly in summer with persistence. But of course, that will not be a recommendation that we will make or something that we will be looking forward in terms of fertilizing alfalfa to improve persistence.
All right. So the next question, have you looked at protein digestibility and efficiency? Because a lot of the protein is lost from the animal.
This is a great question. I'm currently working on a project on alfalfa with Dr. Diwakar Biswas as the animal science department. He's doing a lot of that work. So basically, my measurements are only with NIRS. So I'm measuring total nitrogen in the samples. Now with some of our selections, he's doing some of that work with animals. And we are measuring how much of that protein actually is basically degradable protein compared to just nitrogen. I don't have results yet, but that is work that is coming, and again, it's in collaboration with the animal science department.
All right, next question. Have you focused any on reducing bloat or non-bloating legumes?
No, the short answer will be no. I have current collaborations with the breeding program in Argentina. I know they have been working on breeding for reduced bloat. And I will be looking forward to using some of their germplasm in my breeding program in the future. But we haven't done much work in that sense.
But part of the issue with bloat is low fiber, correct? So if you use right. legumes with grasses, or you use more fibrous legumes like perennial peanut, you have reduced bloat. Is that correct?
I would imagine. I'm not very familiar. So if Dr. Q or Jose would like to chime in that sense.
From my perspective, it's hard to have bloats here in Florida, at least, especially if you have a combination of clovers and other grasses. And during the summer, with Bahia grass and perennial peanut, we never had a problem here for the last 8 years. We've been doing that. Also with clovers and oats, ryegrass, if you have a mixture, typically those legumes are 30% in the grazing. That should be enough to prevent bloating.
Right. Thanks.
I could just make one comment. When I started this project 40 years ago, I said one of my goals was to be able to grow enough legumes in Florida that we worried about bloating cattle. So it's interesting that we're now at the point where people are talking about that.
Thank you.
Yeah, that never happened here. 8 years growing clover, we never had a problem.
I would say for the alfalfa work, I will repeat what I just said. I'm not convinced that we will have monocultures alfalfa around Florida and people will be grazing alfalfa. I think he will be in combination with Bermuda grass or with Bahia grass, and that will most likely reduce the risk of bloating as well.
Or it'll be fed as hay and you'll have a higher level of fiber. If you have questions or you want to talk about some of these ideas on your farm, contact your local county agent and we'll make sure we connect you with the experts that can answer your questions.